how can it be increased or decreased, what things affect it and which organ controls it, and the function of epinephrine or adrenaline based on where you live. Okay, so to start with, this is the famous guy you need to know. Which things are asked the most in paper? Sure. So this guy, William Harvey, is the guy you need to know for this specific subunit. What he did is he kind of tied his hand up and looked at the flow of the capillaries, and his findings were blood flow through vessels is unidirectional, so there is no backflow happening. And then he found out that valves prevent backflow. And the rate of the flow is too high for the blood to just disappear, because before William Harvey, people used to think that once the blood was pumped through the heart, it would go to the organs and it would just stay there, like it would kind of like disappear, it would get used up. And he also predicted that capillaries exist, because back then we didn't really have enough resources to know that there were such things as capillaries. We only knew about the bigger vessels. He predicted it, and he was right, but these are the things that he figured out. Wait, can you hear me? Because someone just said that they can't hear me. Oh, okay, cool. Now, we have the arteries. So you have to know the structure of the arteries. We have, going from outside to inside, we have the so the layers are called tunica. I don't know why, but the outer one is called adventitia, and the way you can think about it is that it's because it's really adventurous. It's kind of like in contact with the rest of your body. Then we have media, middle, I guess, and then we have intima, or the most intimate layer, I guess, because it's in contact with your blood. That's how you can like remember it. So what the structure of it is that in adventitia you have the thick outer layer. It's usually collagenous. It's kind of holding everything together and making sure your blood vessels not kind of falling apart. Your media is mostly smooth muscle cells and elastic fibers. Elastic fibers is really important. Make sure you remember it. The inner layer, though, it's mostly just endothelial cells. So they're a specialized type of cell that kind of just support the body, and they're known to be the ones that just cover everything pretty much. So the most important layer kind of is the media one in arteries that you have to know about. It's because of the elastic fibers. So what's the definition of arteries? Arteries, by definition, are everything that comes out of the heart. You might be hearing that they carry oxygenated blood, which is true in most cases, but don't forget about your pulmonary arteries. They carry the oxygenated blood. They are still arteries, and they go out of your heart. That's why they are arteries. Their structure, though, we talked about that. However, sometimes we have some issues between the elastic fibers and the collagen fibers in the media and adventitia layers. So that can lead to aneurysms. I've put a picture of one of the forms of aneurysms up there, so you can kind of imagine what it is. Basically, a part of your vessel starts blowing up like a balloon, and it can rupture. And if it does rupture, and we're talking about arteries, right, not capillaries, so they're going to have a lot of blood flowing through them, you might bleed out and die. Or you can also not die, but, that's a possibility. So there are risks to it, really high risks. You can also check your pulse using your arteries. So usually any major vessel passing over your bones, if you touch it, you can find your pulse. The most common ones are your wrist on your thumb side, your neck, like pretty much anywhere close to your jaw. And there are like a lot of bony like landmarks on your body you can find your pulse on, but these are like the most famous ones. Yeah, so about do I have anything on that? Yeah, I do. So about the different layers a bit more closely, the externa, as we said, it's just tough connective tissue. Adventitia is also called externa, by the way. Use whichever one you like the most. So it's just connective tissue. It's really tough. Again, it's holding everything together. Intima is smooth because we want to make sure that the blood is flowing through as smoothly as possible. We don't want it to get stuck anywhere. We don't want clots to form. And the thing that makes tunica media elastic is the protein elastin. Yeah, so that's all you have to know for your exam. Like, that's all the detail you have to know. And the reason why tunica media needs to have smooth muscles and elastic fibers is because for smooth muscles, your vessels constrict and dilate. So based on the need of your body, based on your temperature, based on how stressed you are, based on just many different things, your vessels have to change size. The lumen, which is basically the hole of your vessel where the blood flows, it needs to be bigger and smaller depending on conditions, and that's what the smooth muscles are there for. It also just gives it more, strength, because they're your arteries. They have most of your blood. You want them to be strong. We need elastin because when your heart pumps, it's gonna come out, like the blood's gonna gush out with, a lot of pressure. This elastin helps the pressure to stay as the blood goes through it, and it also helps the blood have that same pressure to go from where your heart is, your chest, to your toes. So it has to go quite a long way to go to the distal parts of your body, your fingers, your toes, anywhere that's a bit further away from your heart. So elastin really helps with that. That's pretty much it. Now, you have, for your blood pressure, arterial blood pressure, there's usually two words you use. There's systolic and diastolic. So for, it's usually like a fraction kind of when they give it to you. It's a bigger number over a smaller number. So it's always systolic that has the bigger number. It's your peak blood pressure. It's because when your heart pumps, again, it has a lot of energy and it kind of stretches that elastin, and that elastin keeps the potential energy, for those of you that take physics, throughout until it just kind of reaches its lowest point, which is the diastolic one. Again, we talked about how your lumen has to increase and decrease in size. That's also called vasodilation or vasoconstriction. Fancy terms, you don't really have to know them. For systolic, your normal range is usually 90 to 120. Diastolic, that's usually 60 to 80. This will probably only come up in database questions. You don't really have to know these numbers, but if you wanna, just for extra knowledge, that's like usually the range you want to keep it at. Yeah, so don't worry about the numbers too much. You must be given them. I'm like 99% sure. And they will probably come up in database questions if they do come up. So yeah, that's your blood pressure. Oh yeah, and a way that I remember it is because we also have systole and diastole for the heart. So what you can remember is sisters are mean. So when you have I don't have a sister. I'm sorry to anyone who's a sister, but systolic can mean that you're under a lot of pressure, so everything is tough. And then diastolic, you die. So it's like your lowest, your most relaxed form. Yeah, just a random little thing. I'm gonna check the chat for a second because I can't check it and present at the same time and see what people are saying. Sorry. God, so many people. The names. Yeah, you do need to know the names. Systole and diastole. They are pretty important. They're also in the heart cycle, cardiac cycle. So yeah. Do we have to know the different parts of ECG? I have checked your what's it called? Oh yeah, syllabus. And I've also checked the understandings. You don't really have to know ECG, but I've included it anyway. Because it's not there, but sometimes you're asked it for some reason. I don't really know what's going on with the IB, but I'm gonna. Oh, tunica, intima media. Yeah, those are the arterial structures. You have to know it. And also, I am not recording it, but someone in the audience is. Okay, cool. Anyway, back to the rest of it. Sorry, I'll come back to more questions if you have any. Please move. Why is it not moving? Help. Okay, fine. So we have the capillaries. These are your smaller vessels, okay? So from arteries, that turns into arterioles, and that turns into capillaries. And when it wants to go back to the heart, it's capillaries, venules, veins. Okay, so capillaries by definition are 10 micrometers or smaller in diameter. They're branches of arterioles again. If you remember the arteries, like the structure, you remember how thick and full of everything they were. Now look at the capillaries. They're really small. They're like one cell thick, and they have a really thin endothelium. So again, endothelium was that specialized tissue that just covers the inside of everything pretty much. And they have a protein gel or the basement membrane surrounding it. For this one, you only have to know that they're one cell thick. You don't need to worry about the basement membrane or the endothelium and remembering their names. Now, they have to be permeable, right? They have to be one cell thick. Why? Because they have a lot of exchange stuff happening. So in your lungs, you have to exchange oxygen, carbon dioxide with the blood that's passing through, and you need the one cell thickness to kind of have that happening as fast as possible. You don't want it to go through like a bunch of cells and then finally reach a destination. That's going to make it so much slower. Aside from that, when you bleed, you want your platelets to go out. So they need to be permeable, they need to be thin. The number of the capillaries and how permeable they are, that depends on the tissue. So not all tissues have the same level of permeability for their capillaries. Now, for the lens and the cornea of your eye, you don't really have capillaries. And the reason for that, they're just an exception. And the reason for that is because you need it to be transparent, right? Because you kind of want the light to pass through without any like objections in the middle. So yeah, that's a little cool exception. Now, what are the things that capillaries do? Again, in oxygen, carbon dioxide, they also form tissue fluid or extracellular matrix or interstitial fluid, whatever you want to call it, the matrix. And they also do glucose and waste diffusal. So they have to do quite a lot of diffusion. And yeah, that's pretty much it. There are also benefits to the fact that plasma of the capillaries can leak out. I think I just said the benefits before I asked the question. Sorry, I didn't see that I put it there. But yeah, that's pretty much it. So they help in exchange of material. Now we have veins. Again, veins are kind of just not as strong as arteries because they don't need to be. By definition, they're any vessel that goes to your heart, and it's like quite big enough. Your most famous veins that you have to know are your inferior and superior vena cava. Yeah, vena cava. Sorry, I don't know why I say arterial structure. I meant venous structure. But they have barely any muscle. They have less elastic stuff. They're wider in the lumen, and they have valves. Arteries didn't have valves. These have valves. Why? They want to prevent backflow. So by the time your blood reaches your, I don't know, toes and wants to go back, the pressure, all that little nice elasticity is lost. It's been traveling for such a long time. It has to go, work against the gravity to go to your heart. And it's done some exchanging stuff. It's gone through capillaries and gone back through venules, and now it's in a vein. It's gone, it's done a lot. So it doesn't have that much pressure anymore. These back valves help prevent backflow. So the way your blood goes back to your heart, basically from your toes, and works against gravity. Well, some of it is definitely the pressure. It still has some pressure, but the rest of it is because your muscles, as they kind of flex and extend, they put pressure on the veins because veins are not strong, so they can just easily kind of fold, I guess, and that pumps the blood back up. And as it goes back up and wants to go back down because gravity exists, the valves close, and that prevents backflow. And that also helps not form any clots in your legs, because it has to work against gravity. But if you are still for really long times, then you're old and you are overweight, or you have like high cholesterol and like everything else, you might get clots in your legs. So that's why many people try to like get up and like stretch every once in a while. Yeah, so we talked about that. Yeah, so valves try to ensure unidirectional flow no matter the muscular and postural changes that you might have. That's pretty much all you have to know for the veins. Yeah. Okay. Next one. Yeah, okay. So now we're going to try and draw a heart. So I'm going to let those of you that want to fetch a pen and paper. And it's okay if you don't want to. Yeah, I'm going to let those of you that want to do that. And I'm going to check the chat in the meantime. God, my laptop is being so slow. What's the difference between arterioles and arteries? Arterioles are just small arteries, so they're less thick, they're smaller in diameter, and they're a branch of arteries. So your arteries give rise to arterioles, and then they give rise to capillaries. Yeah, we're gonna try and label a heart. Okay, will we ever get asked to draw a heart? Because I would have assumed I've just been asked to label it since it's so complex. Yeah, you can be asked to draw a heart. You can also be asked to draw kidneys. There's a lot of stuff that they can ask you. Usually this is for, the last bit of paper 2, I think, when you choose 2 out of the 3 questions. That's usually like one of them. The kidney is not included in SL. Sorry, I was just giving an example. Okay, cool. I'm assuming people have their pen and paper. I'm gonna wait for Cookie Dough and I don't know to finish typing. Yeah, okay, cool. Yeah, we are doing SL right now, don't worry. Sorry, I should have clarified. Okay, now, draw a heart. Please draw your little schematic heart, like a little thing, and we can start labeling it together, and you can know what things you have to include and kind of know from like names and like parts and everything. Okay. So I'm assuming you have something like that. Sorry, I know it's really grainy and blurry. But yeah, I'm gonna assume that you have something like that by now. To start, we're gonna uncover some bits. So I'm gonna ask you, do you know what that part is? So a tip is, because this is kind of like, well, not mirrored, but it's reversed. So your right side isn't gonna be that heart's right side, is to look at the ventricle. If it's a correct image, your left ventricle is usually thicker. So if you forget which side you're looking at, you can always look at the thickness of the muscle for your left and right ventricles, and you'll see that the left one is usually thicker. Sometimes that's not the case, so obviously don't always rely on that, but it just helps if you can see the difference. Right, so that's going to be your right atrium, and then right below that, the blood goes to the right atrium, goes through the atrioventricular valves, and then we have the right ventricle. And now, on the other side, again, we have this little thing up here that's going to be your left atrium, goes through the atrioventricular valves, and you have your left ventricle. Now, from your left ventricle, the blood gets pumped out and it goes to your aorta. That's going to be your oxygenated blood. So your left side only deals with oxygenated blood. If it doesn't, something's wrong. So it goes through the aortic valve and it goes to the aorta. Okay? So you have to be able to label them. And then from your right ventricle, now the blood that is deoxygenated, it comes to your right atrium, so your right side only deals with deoxygenated blood. And the main veins that carry this blood to your right atrium are going to be your superior and inferior vena cava. So they're going to come to your right atrium, deoxygenated blood, go through the atrioventricular valve, go to your right ventricle, and get pumped out to your pulmonary arteries through your pulmonary valve. So your heart has 4 valves. 2 of them are going to be atrioventricular, and 2 of them are pulmonary and aortic valves. So the valves that take the blood to your other circulation, basically your systemic or your pulmonary circulation. So that's your pulmonary artery. And the atrioventricular valves, one of them is tricuspid, one of them is bicuspid. I'll show you in a second, but the tricuspid one, it's usually your right side. The bicuspid one is going to be your mitral valve. So your left atrioventricular valve is also called your mitral valve. So it's a really important valve. Make sure you know it, because your left side kind of deals with your entire body. So it's important and it's strong compared to the right side, which only has to take it to your lungs, which are right next to your heart. And the part that separates your right and left ventricle is called your septum. So it's just the muscle trying to separate your kind of the different segments of your heart. Now I'm gonna check the chat, and sorry, I'll go back so you can also have that in there. And if you're labeling, if you're actually drawing with me and labeling them, you can use that. Okay, 19 different. Okay, wow. Okay, diagrams are really important. So if I don't have drawing skills, you don't need drawing skills. I used to draw my hearts like acorns. You're fine. As long as you have the basic structure and division. Yeah, exactly. You can literally draw boxes. You don't need to draw like anatomy atlas hearts. Exactly. Yeah. Oh wow, that's a nice mnemonic. Thank you. Cool. So if no one has any questions, I'm gonna continue. Yeah. So we're gonna talk about atherosclerosis, my favorite thing ever. So that usually happens in coronary arteries, and it will result in your coronary heart disease or ischemic heart disease. Atherosclerosis, let's look at the picture and see what it is. It's basically plaque buildup, and it can be fat, it can be calcium, it can be anything. So basically your tunica intima layer, that's going to have some fat on there. And what is going to happen is your blood's going to be obstructed the flow. So you're going to have blood clots forming, and your lumen is going to become narrower and narrower and narrower. And at one point, blood's just not going to pass through in a rate that's enough for your body to be supplied enough. So that can happen in many different vessels. Usually it happens in coronary arteries, as I said. So those are the arteries that supply your heart. So that's going to be bad. Your heart needs its oxygen, its glucose to kind of work, and not having enough blood flow won't allow that. So your heart tissue will die. So what's going to happen is you get atheroma next to your endothelium, so that your tunica intima. Usually it's because of high levels of LDL or lipoproteins in your blood. And you will all, especially at this age of fast food and like fatty foods, have some of this by the age of 10, the ripe age of 10. So don't think you're safe. I don't mean to scare you, but like it's pretty common. It's normal. It can lead to anoxia, angina, and tachycardia, which are fancy words for lack of oxygen to any tissue or your heart pain, and a faster heartbeat. Things that put you at risk of getting atherosclerosis, CHD or IHD, or coronary and ischemic heart disease, are high cholesterol, diabetes, obesity, a bad diet, and a high blood pressure. So these are things that put you more at risk. And this can lead to a myocardial infarction or a heart attack, and death of some of your heart's tissue, muscle tissue specifically. So that's like one of the things that you need to know a little bit about. You probably, I haven't seen many questions on this before, but you never know. It's like one of your understandings. It's part of your syllabus, so do know it. Now we're going to talk about the electrical conduction of your heart. So as you know, your heart is myogenic, it's autorhythmic, the way it beats. So your sinoatrial node, or your SA node, is going to have the fastest rate of spontaneous beating. So your heart is spontaneous. Nothing like kind of tells it to beat. It's not like the rest of your body where kind of like a nerve has to go to a muscle, tell the muscle to squeeze, and then the muscle is going to squeeze. No, we don't have that. That's going to take too long. We don't have time for that. Your heart's going to generate its own beating. SA node, or as you can see here, that thing, it's in the right atrium. It's close to the part where your inferior and superior vena cava enter. That initiates every heartbeat. And then you have your SA node traveling to your AV node or atrioventricular node. It's going to take a while, and by the time it does that, you're going to have the whole entirety of your right atrium and some of your left atrium getting the note that they have to squeeze. Okay? Now, here we can look at the electrical conduction pathway of the heart. So we had the SA node, right? And then that's your AV node. So you have an internodal pathway that you don't need to know about. You don't need to know what the internodal pathway is. This is just to like help you understand. So the heart, the electrical stuff goes from your SA node to your AV node. It also goes to your left atrium because both atria have to contract at the same time. And then after it goes to your AV node or atrioventricular node, it also has to move down your heart because your atria have contracted. The blood has left the atria. It's going to fill up your ventricles. And then when it does fill up your ventricles, they have to squeeze the blood out at one point, right? So we have the bundle of His, and then we have the left and right bundle branches because one of them supplies the left side, one of them supplies the right side. Then we have the Purkinje fibers, and they're kind of like just smaller fibers that supply the apex of your heart and just tiny ones for the outer part of your heart. And that's how your right and left ventricles squeeze. So just to recap, SA node, AV node, bundle of His, right and left bundle branch, Purkinje fibers. Okay? That's pretty much all you have to know. Ignore how complex this looks. I know it's a lot of branches and a lot of labels, but it's really just that. And your SA and AV node, they don't contract at the same time. They have a little bit of, kind of gap in between for the blood to leave your atria, go to your ventricles, for the valves to close and prevent backflow. And then your ventricles can squeeze it out. Otherwise, you would have some problem with the rhythm of your heart. So yeah, the way it's all spaced, they're mostly concentrated in the right atrium. So SA node was close to where your SVC and IVC, superior and inferior vena cava, entered your right atrium, and your AV node is closer to the septum, the bit that separates the two parts of your heart. And it is a bit closer to your left atrium, but it is still in the right atrium area. Okay, before we start the cardiac cycle, I know that's a lot, so let me check the chat. SA is the first beat we hear, right? And the AV is the second beat. Yes, that is correct. Oh my god, wait, did Git draw something? Let me see. That's actually so nice. I wish I could pin it in this chat, but I can't. So I'm just gonna encourage everyone to go see that. That's really nice. Yeah, LDL is the bad one, but you don't really need to know which one it is. Just know that high cholesterol, they're not gonna expect you to know if it's LDL or HDL. They're not gonna care as long as you know. Okay, what's it? Okay, cool. I can draw a really, really shit diagram that I'm pretty sure anyone, even a 5-year-old, can also draw and send it in the chat later. Oh, thank you, Git. That's really nice. So SA gives signal to AV, yes. So SA is basically what causes your atria to contract. AV is gonna help that same signal that helped your atria contract propagate through the rest of your heart, through your ventricles, and then cause your ventricles to contract. So we have the left and right bundle branches and the Purkinje fibers that cause your ventricles to contract. SA contracts your atrium. AV is kind of a connection point. The bundle branches, Purkinje fibers, and the bundle of His are the ones that help your ventricles contract. Okay, great. So is the cardiac cycle basically no? That is unfortunately that's just your electrical conduction of the heart, SA and AV. The cardiac cycle is kind of no, but cardiac cycle is the easiest part. Electrical conduction is the worst part, I promise. Okay, come on, let's do this. It's really easy, I promise you. we've kind of done it already. So we know that the vena cava they drain into the right atrium, and we know that the right atrium and the left atrium, they both contract, right? They both have blood in them. Right atrium is going to have deoxygenated blood. Left atrium is going to have oxygenated, because left is oxygenated. They both contract at the same time-ish, and that's called your atrial systole, because remember, yeah, systole is because it's just contraction, okay? Fancy word for contraction. Then you have your atrioventricular valves open, because valves open in one direction, and the pressure is so high in your atria that will cause the valves to open. The blood will push through, go to your ventricles. Your semilunar valves, or the valves that have the blood leave the ventricles, they're closed because you want the blood to remain in your ventricles, and the pressure is not that high in the ventricles yet. So the ventricles are going to fill up, and then when they fill up, the atrioventricular valves are going to close. So remember, your left side was your mitral valve, your right side was your tricuspid or your atrioventricular valves. They're going to close once your ventricles are filled up. And they're going to prevent the backflow of the blood that your heart just tried so hard to push from your atrium to your ventricle. And then your ventricles are going to contract. So that's called your ventricular systole. And the atrioventricular valves, they don't open. I don't know why I put that. But your semilunar valves, they open. The blood leaves through pulmonary arteries and aorta. I'm going to fix that. Yeah. So then the blood leaves through the aorta and the pulmonary arteries, and the semilunar valves close and prevent backflow. So that's your ventricular diastole. So your ventricle is just going to be chilling. Yeah. So let's do this again. We're going to have the oxygenated blood in your right atrium, oxygenated blood in your left atrium. Atria are going to contract. You're going to have your tricuspid on the right side, mitral on the left side, or your atrioventricular valves to open because the pressure buildup has happened. Your blood's going to gush through and go to your ventricles. Your ventricles are then going to be filled up with blood. Your semilunar valves are going to remain closed. Your atrioventricular valves are open because you want the blood to go from your atria to your ventricles. At one point, when the ventricles are filled up, your atrioventricular valves are going to get closed again to prevent backflow because your heart doesn't want to try too hard. And then, once you have that, your ventricles are going to get contracted, your blood's going to leave the ventricles, go through the semilunar valves, go through the pulmonary arteries and the aorta. So from your right ventricle, it's going to go to the pulmonary arteries. From your left ventricle, it's going to go to the aorta. And it's just going to leave. And once most of the blood is pumped out, your semilunar valves are going to close and prevent backflow, because again, your heart doesn't want to do much. You don't want the blood to go back to your ventricles and then fuck up the whole cycle. So that's what you have. When anything contracts, that's your systole, and when anything is relaxed, that's your diastole. Okay? Now, let me see if anyone has any questions. So the valves act as a gate, yes. Atrial diastole is basically when the blood goes to the ventricles, right? Yes. Well, atrial diastole, not really. It's when they get filled up. Atrial systole is when the blood is getting pushed to the ventricles. Diastole is usually when nothing is contracting, so that's going to be when your atria are getting filled up. But after they're in the ventricles, yes, that would be atrial diastole. Did I say both have oxygenated blood? Sorry, that was a brain fart then. Why do the ventricles need to be 70% full for the contraction in systole? Because why would you pump with an empty ventricle? Your heart doesn't want to do more, you don't want to put in too much effort. You need some blood to be able to pump it. And also, if you don't have enough blood, your ventricles can only contract to a certain extent, right? If there is not enough pressure to make, that area, if there's not enough blood to make enough pressure in that area, then your semilunar valves won't open and let the blood out. So if you don't have enough liquid inside, then the whole system's not going to work. Okay, so many people are typing and I'm scared. Oh, it can be. It's just that when it's too full, it's going to be harder for your muscles to like do it. So yeah, 70% is the minimum. It can be 100% full too. Okay, I'm gonna continue this session. If you have any questions, oh, does heart get empty? Does heart get emptied when the blood leaves? No, some blood always remains in all different chambers of your heart. But it's just a little bit. And it's like it's moving so much it won't clot usually because it's like 60 beats per minute, right? So your heart's always in motion. Even if your ventricles aren't contracting, your atria are, and that's going to shake up your whole heart kind of a little bit. Atrial diastole is when your atrium fills up, yes. Systole, atrium pumps blood to the ventricles, yes. Ventricles fill up. Yeah, when they have pumped and when they are filling up, they're going to be relaxed, right? So it's going to be diastole anyway. Yeah, you are correct, Adelina. Which vessel provides oxygenated blood to heart? Coronary vessels. There's quite a few of them. You have your right, left, marginal, circumflex. You don't really need to know them. If you do, though, if you want to know them, there are pictures on Google. Just coronary arteries. That's all you have to know. Yeah, and as Git said, it is quite continuous. Okay, moving on. Now that you know the cardiac cycle, you can start to. Oh wait. Okay, yeah. Little break. You can see that all your different valves have 3 bits, but your mitral has only 2. Your mitral was your atrioventricular valve on the left side, and that's a really cool diagram that I'm really obsessed with, and I wanted you to appreciate it as well. You don't need to know it though. Okay, now that you know the cardiac cycle, you can appreciate this. It is in your book, you might get it, and I know it looks disgusting, trust me, but it's really easy if you think about it. So you only have to know when your mitral valve closes, when your aortic valve opens, when your aortic valve closes, and when your mitral valve opens again. And that's pretty much it. So let's go through it step by step. So when the lines go up, that means there's higher pressure. So when your mitral valve closes, what does that mean? That means that the blood has entered your ventricles, specifically your left ventricle, and it is now preventing backflow. So that means your atrial pressure is going to be at its lowest because your ventricle has pumped, the next cycle has begun, and it's now getting ready to pump. So there's not enough pressure in the atria anymore. There's not much pressure in the atria anymore. And the ventricular pressure is going to start increasing because it's getting filled up more and more and more, and it's going to start to eject, right? And your aortic pressure, it's going to be, pretty neutral at that point. You don't really have much happening. But then the second your aortic valve opens, that means your ventricle has contracted. Your ventricular pressure is going to skyrocket because it has fluid in it. It's getting squeezed. All the blood's going to leave it, but it's under a lot of pressure right now until your semilunar valves or your aortic valve opens, and that's going to cause your aortic pressure to also increase. The second your aortic valve closes, that means the blood has left most of the blood has left ventricle. It's now in your aorta. The aortic pressure is going to have a little dip because it was getting a lot and potential energy, and now it's not getting more. So that's going to have a little dip, and your ventricular pressure is going to keep decreasing at a steady, really high rate. So it's just going to fall really fast and wait for the mitral valve to open and get more blood from the atria. So that's pretty much it. I know it looks really disgusting when you look at it first and really, really, really hard to kind of like wrap your head around it. But if you just think about the different stages of the heart where most of the fluid is and where things are contracting to make the pressure even higher, you can kind of get this, like, you can understand it pretty easily. So, yeah. What else? Oh yeah, and systole and diastole again. You can kind of think about that as well. There's also isovolumic contraction and isovolumic relaxation. So isovolumic basically means that the volume stays the same. And isovolumetric contraction, though, it refers to the brief part where all 4 valves of the heart are closed at the same time. It's really brief, and relaxation is when all 4 are kind of open at the same time. So yeah. Also isovolumetric contraction is when the ventricles are just beginning to contract, so they're getting ready. And yeah, relaxation is when they have just contracted and they're done. That is a better definition. Yeah. So contraction is when they're getting ready to contract. Isovolumic and isovolumic relaxation is when they've just contracted. You don't need to know it though. I'm just explaining the diagram to you a little bit. So let me check the chat. Oh god, my Discord broke. Hello. Why does this shit always happen to me, man? Okay. Can you explain the William Harvey experiment? I can go over it again at the end of the session. It was at the start. Which vessel? Okay, yeah, we've done that already. Bicuspid / tricuspid difference. Oh, okay. Oh god, my Discord is acting up, guys. Sorry, give me a second. Again, you're not supposed to know it, but I've seen it come up in like some papers. I don't know why, but we can just do it for the sake of doing it. So firstly, you have your P wave. It's labeled on the left-hand side. You also have the key on the right side. So you have your P wave. That's when your atria contract, also known as the depolarization of the atria. Then you have your Q, which is your ventricles basically getting ready to contract. Your R wave, that's the little spiky thing, that's the depolarization of the ventricular myocardium, or when your ventricles do contract. S wave, that is basically when the blood has left your ventricles, kind of. And T wave is ventricular repolarization, or when your ventricles relax. Now you can kind of put this and this together, and guess what happens? So we have the isovolumic contraction. That's when your ventricles were ready to eject. That will be your Q little thing. And we also had the isovolumic relaxation. That's going to be your S thing. T is when everything is relaxed, and you also have your PR, ST, QT, and all that. You don't need to know any of that. As long as you know P is your atria contracting and R is your ventricles contracting, that should be enough to get you through IB. But yeah, that's an ECG, since I know one of you asked for an ECG little thing. Yeah. And thank you. I'm going to go through William Harvey at the end, but if you do have any questions, let me know now. Okay, do you have any questions? Should I go through William Harvey? Explain the control mechanism of the heart rate. How do we write this for 7 marks, and is a diagram required? Some people have attempted it. I'm not sure how to answer it. I didn't talk about, okay, yeah. We can definitely do that. So, the control mechanism of the heart. So firstly, you can talk about how it is an algorithmic myogenic thing. So your SA node going through your AV node, going through your bundle of His, your left and right bundle branch, and your Purkinje fibers. So that's how your heart rate, I will share the PowerPoint. That's how your heart rate kind of works. And then you can talk about how adrenaline increases your heart rate. You can talk about how being stressed increases your heart rate. You can talk about how the medulla in your brain is basically your heart rate control center. You just have to talk about how the action potential kind of propagates through the rest of your heart, the contraction, the control, yeah, that's pretty much it. So just to summarize your essay and AV, guys, I'm gonna pretend I didn't see that and I'm gonna delete it, but rule 13. Anyway, yeah. Do what you want, just don't say it in the server. I support all piracy, but we don't want to get in trouble. Thank you. You don't have to talk about the sympathetic and parasympathetic nervous system. All you have to do is adrenaline, epinephrine. That is going to make your heart rate go faster. And you also have to know that there are two nerves that control your heart rate, two things coming from the medulla in your brain through your spinal cord to your heart. And that's one of them increases your heart rate, one of them decreases it. The rest is just propagation. How do you generally frame paper 2 long answer questions? What do you mean by that? Like, how do you outline it? Like, what's your structure? The bundle of His is the connection from your AV node to your septum to the rest of your heart. So it's like a little bundle that takes that propagation to your ventricles from your atria. I didn't really format anything. I just wrote whatever that came to my mind. I kind of blurted it out. Obviously, don't be like me. If it's like a hard question or like anything human physiology, then you definitely have like an outline. It's like step by step. You're kind of telling a story, so you can definitely follow that storyline as you're outlining your answer. But other than that, like you just have to do step by step, I guess. Yeah, it does transfer the stimulation to the ventricles, yes. Propagates the rest of it, yeah. I'm gonna be real, I just wrote everything that I knew in paper 2, which is really dumb and I shouldn't do it, and I'm saying you guys please don't do it either, but that is one way you can like structure your paper 2 answers. Just write anything that's like relevant to the question, because you can just get marks. So it goes SA node, AV node, bundle of His, and then you have your right and left bundle branches and your Purkinje fibers, and yep, those do your ventricle contractions. How did you study for paper 2? I kind of like used to read the textbook throughout the 2 years, and then at the end I did Bio Ninja and I did some like past paper questions, pretty much memorized the mark scheme and kind of like figured out what they wanted from me, because it's not just about your understanding, it's also about what they want from you, what they want to see. So mark schemes are your best friends. I didn't use Cheryl Hickman, sorry. What grade did you get bio? I was HL and I got a 7. The IA, I mean, yeah, it's like 20%, isn't it? So it kind of, yeah, obviously it is important, but it doesn't mean that you don't have a chance to do well if you did bad on your IA. Or if you did really good, don't get too proud. There is still a chance to like have your grade go down. So all the papers are just as equally important. Database question feels hard. How to do it? You just have to like know how to answer it. I can kind of go through how to answer database questions actually. Then won't the mark scheme depend on the examiner corrections? Because my teacher says don't memorize the mark scheme. Your teacher's right. You have to know the actual thing. Like you have to know the content. You just have to know how to structure your answer the way the mark scheme kind of looks at it. So you have to have your golden nuggets, as a friend used to call them. You have to like you have to like know like what you're saying. You just have to also know what they're looking for, so you can change it in that way. No, I'm not. I'm really dumb. I used to get fives in bio. In paper one, the topics are equally divided. Yeah, I would say they're all equally divided. What topics did you find the most asked in paper two? It's usually topic 8, 6, and 11. Like if you look at the long answer ones at the end, which count the most, I guess. Usually topics that they can ask you to explain something. So like muscle contraction, nerve, like action potential, and like propagation of that. Kidneys, heart, I don't know, clotting system, your antibody formation. Yeah, that's like usually your longer ones. The mark scheme is created by the average answers given by candidates. No, the questions have their own mark schemes, but you do have like the and or tweaks that are added to it because of what the candidates have said. So the mark scheme isn't always like, are you gonna do HL topics? I'm gonna do the kidney too next week, actually. I score 4s all the time. It is so possible to reach a 7. Just do Bio Ninja and like do a lot of past papers. I promise you'll get a good grade. First, list down like keywords or vocab based. Yeah, I mean you can do that. I personally don't work like that, but that is a good way. Topic 8, topic 11, topic 9 as well, actually, for plants, like the way they grow and the way source to sink stuff, that like how the phloem and xylem, how movement happens, that's also something that's asked in paper 2, I remember. Yeah, 6 and 11 are usually connected, 2 and 8, yeah. Oh god, I hate topic 8. The pain it brings me. I'm May 2022. Why? Plants are really nice. They're actually like way better than topic 8 or like, I don't know, antibodies. Okay, can we have exam strategy session? Sure, I'll try. Okay, do you guys want to like look at how you can explain a graph? I'm gonna try and like say that for database questions. Okay, what do I want to search? Oh wait, I forgot to add bio. I feel like I shouldn't do this. Let's just go for graph snails. Yeah, we can go for the graph snails IB. If you know, you know. Okay. Why did I choose this one? I actually hated this question. Okay, we're gonna look at that and the one at the top so we can know both of them. So open image in new tab and also this one. So you, oh god, that's so small. So you can ask questions and like explain this. And then you have to find reasoning for it. So I can't obviously talk about reasoning because I have no idea what's happening here. But so what you have to do is you have to say that you have the familiar and unfamiliar types / F1 or U1, I guess. And what you have to do whilst you're explaining is you have to say what happens to it. So the black one goes from 0.5 to 2 in the span of X many years. I don't know. It's a random graph. And the white bit, the transparent bit, remains almost the same, so it has a really slow increase compared to the black one. That's something that you have to say. When you're asked to explain a graph, you have to state the numbers, the amount of time, the labels. When you're asked to describe it, you only have to state a trend. Obviously, look at how many marks it's like, you know, getting allocated. So if it's like 4 marks, you obviously have to say way more. If it's like a mark, then you only have to say the black one increased. If it's 4 marks, then, you know, the black one did X and Y, the white one did X and Y in the span of time, in the span of like that many years, that many weeks. It can also be observed that like the increase was exponential. I mean, you can't see it here, but if you do have a graph, you can say it was exponential, linear, I don't know. You can also like give it some adjectives like that. You just have to kind of like really be really descriptive about it. This one is, I think this is actually like an IB graph. We had the different snails with bands or no bands, the yellow shells. So you could see that most of the, I don't even know how to pronounce that, but the wood ones, most of them had like 40 to 100% of them had one or no bands. So most of them had less bands, and not many of them had yellow shells. But the other ones, most of them had yellow shells, so up to like 50% of them had more than one band. And the percentage of them having yellow shells was like quite all over the place. So a big range. So you can also talk about the range of it. You just kind of have to go through like the different stuff and analyze it. Yeah. This graph is really disgusting to analyze. I know, I hated it. I'm just gonna trust Git and not read that whole paragraph. Do we need to know the nature of science? I'm not gonna lie. I don't know what a nature of science is. I don't know what that is. What is it? I don't know what it. Why are you crying? Oh, are we talking about myelin sheath? Oh yeah, we are. Is that HL or is it SL? If you have a next session, can you cover inheritance and immune system units, please? I'll try. Guys, I also have my exams in like 40 days and I'm in med school and I am so close to failing. So like, I'm not gonna make any promises, but I'll try. I'll really try. Thank you. Is there neurons in SL though with sodium and potassium pumps? You do have some neurons in SL, yeah, but most of it is H. Like the hard, complicated parts are in HL. So for SL, it's like the basic understanding, kind of. Good luck to your sister too. We haven't finished. This just made me feel depressed. Please do try to finish the syllabus on your time. Thank you. You too. Yes, I do regret it every day. Thank you, Alicia. And we talk about William Harvey. Yeah, sorry, I forgot that. Let's go back to William Harvey. Okay, yeah, William Harvey. Oh, shit. So William Harvey, what he did was he basically tied a little ribbon around his arm. He was feeling coquette, and he tried to experiment what would happen if he, like, didn't let the blood flow and let it flow back again. So you can also try it with like your fingers. If you like push on the tip of your fingers, you'll see that it will go a bit lighter in color and then it will turn back to its natural color. Usually easier to see it in people with fairer skin, but you can just see it, I guess. If you like push a little bit, you can see that it goes lighter and then blood fills it again. That's what he did, but like on bigger vessels. So he figured out that blood flow is unidirectional, so it doesn't just go anywhere it wants. You can't have blood going back and forth in one vessel. He also figured out that valves exist, they prevent backflow. I don't know the details. You don't need to know the details of how he did it. You just need to know he tied something around his arm and he figured that out. And he also, in that time, people used to think that blood used to just disappear after it left the heart. He figured out that it doesn't disappear, that the rate is so much, the flow is so much that you can't just have the blood go poof. So the blood does eventually return back to the heart. He figured out that veins exist. And he also predicted that capillaries existed, but they didn't really have enough equipment to actually figure it out at the time. So he died before capillaries actually got discovered. But he predicted it, so kudos to him. I haven't finished. Time for us. Sucks, but better than last year. Oh god. I mean, if you know your HL syllabus, then you probably know the SL syllabus, but still try to finish it. If you don't have time, there are like cram stuff kind of on YouTube, and also BioNinja can really help you like know what you have to know. It's definitely a bit harder to understand what BioNinja is referring to if you don't know the whole content and you're just knowing the high yield content, but it still works. What are my thoughts on BioNinja? I love BioNinja. We didn't do options. I know, I've seen it. It's actually really ugly, the new BioNinja, but it's okay. Like it still works. I think it is reliable. Like if you have learned stuff before, BioNinja is basically all the understandings and everything in the syllabus. So it's everything you have to know. It is reliable. Oh yeah, you can self-study options, but it's a bit of a gamble. Okay guys, if we're done and you don't have any questions from me, you can continue your chat in the M24 bio chat. If you do have any questions from me, you can ask me now or you can ping me later on. So thank you for joining. I really appreciate all of you and good luck with your exams. I'm sure you're going to do amazing. So bye for now. Take care.